everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today we're talking to Shannon Macauley, who is Associate Professor in the Departments of Physiology and Pharmacology and the Sticht Center for Healthy Aging and Alzheimer's Prevention at Wake Forest University School of Medicine. Shannon's lab studies central nervous system diseases and their mechanisms with an emphasis on Alzheimer's disease, at least right now.
Yes.
And some of her recent experimental work has been on the relationship between Alzheimer's disease pathology, meaning tau and amyloid beta production and accumulation, sleep patterns, type 2 diabetes in laboratory animals. She studies all that using a combination of biosensors, local field potentials, microdialysis to track local concentrations of glucose, lactate, glutamate, insulin, A-beta, and tau in the hippocampus over the sleep-wake cycle in old and young animals, and ones with A-beta pathology and ones that don't, and during hyper and hypoglycemia. And that's what we're going to be talking about today.
Sounds great.
So hi, Shannon.
Hi.
Welcome.
Thank you.
We also have our own Melanie Carless, who has also got a research program on metabolic disorders and their relationship to neurological diseases. Hi, Melanie.
Hi, Charlie.
And a PhD student in our department who's working in Melanie's lab, Uchit Bhaskar. Hi, Uchit.
Hi.
So, Shannon, to get us started, we know that type 2 diabetes patients are much more likely to suffer from Alzheimer's disease, and this has implicated glucose metabolism in the etiology of the disease. But of course, it's difficult to sort out cause and effect in the many dimensions of human disease. And could you give us a little summary of the experimental approach you're using to identify what's cause and what's effect in the changes in brain glucose, lactate, insulin, sleep, aging, diet, and Alzheimer's-like pathology in experimental animals?
Sure. Yes, we try to study all of the factors. So as you mentioned, in humans, it's very difficult to know because most of these diseases are unbelievably complex to know what aspects of these diseases are really driving pathology of another disease. And this even gets more complex because as somebody who studies Alzheimer's disease, that typically when somebody comes to autopsy, you don't even see that they only have Alzheimer's disease in the brain. They have a lot of other types of dementia and other types of protein aggregations and things like that. So what mice allow us to do is really simplify the complexity of the diseases. So Alzheimer's disease is normally made up of neurofibrillary tangles and amyloid plaques. And we have mouse models that either get plaques or they get tangles. So right then there, we can disambiguate whether some of the metabolic changes that we want to know about are altering either a form of the disease or either arm of the disease differentially. And then similar with type 2 diabetes, it's a very, very complex disease. It certainly affects our metabolism, but it also affects our vasculature, it affects our sleep, it affects pain levels. And so again, if we start to isolate different aspects of that disease by looking how your blood sugar becomes elevated in type 2 diabetes or your insulin becomes dysregulated, we can then go back and look in our animal models that only get amyloid plaques or neurofibrillary tangles, how those things change.
It seems to me another dimension of it is sort of blood levels of things versus brain levels of things. And so it's really they're not always exactly the same. Sometimes they are very closely related.
Right.
Sometimes they're wildly unrelated.
Right.
And so your approach is to actually stick stuff into a particular part of the brain,
right?
The hippocampus.
Yeah, and so we've taken a combination approach. So for a long time, you can do this in humans, you can do it in different animal models, but you have the ability to do what's called glucose clamps, for example. And this way you can actually insert really small tubes or catheters into different veins and arteries. This allows you to infuse different substances. So we do, raise blood sugar with glucose, or we raise insulin with insulin, or you can actually even deliver drugs this way. And why we do this, we combine this with another set of experiments where we can actually do in vivo microdialysis. And what that means is that we can take regions that are either vulnerable or prone to pathology and stick a microdialysis probe in. And what it actually acts like is kind of like a little soaker hose that takes up all of the fluid from in between the cells of the brain or a specific brain region. And you can actually test different levels of metabolites, different levels of proteins, and see how they change in real time to whatever change in systemic metabolism or biology that we introduce into the animals. So unlike doing a treatment that lasts, 3, 6, 9 months in an animal, and then just looking at a very static time point at the end, our preparation allows us to look very dynamically about how things change in real time in different compartments. So thinking about what's happening from the neck down versus the neck up.
Maybe this is a little nerdy, but I'm wondering is everything done with microdialysis, all with one microdialysis unit, so that you're pulling a bunch of fluid out and then you find glutamate, you find the glucose and the lactate and everything all from one sample?
Yeah, so we have a variety of different approaches depending upon the question that we're asking. So microdialysis is kind of great because you just get that interstitial fluid out, and then you can assay that fluid in a variety of different ways depending upon the question you have. So we have separate assays that we then take back into the lab and look at glucose, lactate, or whatever. We also then have ELISAs where we can actually look at very small changes in concentrations of amyloid beta or tau in that interstitial fluid, and then again, correlate it back to changes in metabolites. The biggest limitation with this is that you have to collect the fluid at a really, really small flow rate. And so over the course of an hour, you only get 60 microliters, so a very, very small volume. So you really want to a priori decide what you want to, to look at. But then we have other ways that actually use biosensors, and those are specific to a specific metabolite. And so we can implant those in the same way in the same region, but they actually use a change in voltage and an oxidative step to measure changes in glucose or lactate, but each probe is specific to one analyte. But the thing that's good about that is that temporally, it's a very quick reaction, so we can get better temporal resolution than we can with microdialysis, but you can't look at as many things as you can with microdialysis.
And then these measurements can all be made in an animal that's walking around doing
That's
things.
right,
So
yeah.
And you can follow the same
Yes.
animal through the day-night cycle, for example.
That's right, yeah. Yeah, and so we also even have other techniques in our lab where we use small animal neuroimaging, where we can actually, and this just gives us reads of, cortical blood flow and hemodynamics, very similar to what people do with functional MRI. And so we can look at, we know that when a certain part of the brain is firing, it recruits metabolites and things, recruits blood flow to that region, and we can actually look at oxy and deoxygenated hemoglobin as a metric of neuronal activity. And so in those animals, we also train awake because we don't want to have the animals anesthetized because that interferes with the metabolism of the brain, as well as obviously the intrinsic, basal firing and things like that.
So maybe we could get a little bit of the results, especially regarding glucose and lactate and A beta.
Yeah, so we started out by asking a very simple question with type 2 diabetes. We knew that type 2 diabetes is kind of defined by this chronic increase in blood sugar, blood glucose levels. We know that insulin levels typically rise and then fall as the pancreas becomes taxed. And so we just wanted to ask the question of, if you raise blood glucose levels or you raise blood insulin levels, do they change the levels of amyloid beta in the brain? And we know that within the brain is once you have that amyloid beta in that extracellular space, and if it's not cleared right away and the concentration increases, then it starts to aggregate and form those amyloid plaques, which is the hallmark of Alzheimer's disease. And so what we found is if you increase blood sugar, then we increased A beta levels into the brain. And when we looked at this, it looked like it was done by making the neurons just more ready to fire. In most of my talks, I include a picture of my daughter when she was 8 years old, and we gave her a bucket full of cupcakes. And if you've ever seen a kid like gobble up cupcakes and their eyes go wide and they run around in circles, in essence, that's what happens to your brain, is that this sugar from your blood vessels gets into your brain, it freely diffuses and just makes your brain more excitable. When your brain's when your neurons are firing, it makes more A beta. Too much A beta aggregates, and then you can start to go down this Alzheimer's cascade.
So how come every kid with a cupcake doesn't end up with Alzheimer's disease?
Because kids are resilient and buffered. And because they'll go do that, they'll go run in circles and be bananas, right? And then, in that type of activity, whether it's exercise or any of other factors, can actually increase A beta clearance. So the aggregation of amyloid or A beta into amyloid plaques is always the seesaw of whether you're producing too much. So we've shown that, high levels, high elevated blood glucose can drive A beta production. But as long as you have an equal clearance of A beta, then you don't have aggregation. So in a young, healthy brain, this can be occur via sleep, right? You go to bed, you have this kind of bulk flow clearance mechanisms in play, and then you probably don't have aggregation. But as we get older and our vasculature doesn't work as well, we're not as active, we're more sedentary, we eat poorly, then you really increase probably the production, decrease clearance, and boom, you have aggregation.
It sounds like that's the link to sleep.
Yeah.
Is this clearance idea?
Clearance and production. So when you're awake and your brain's firing under normal physiological conditions, you actually make A beta and you release A beta from the neurons. But again, as long as things are moving properly, then you just clear it from that extracellular space. But then you have the added bonus of sleep. And so not only during sleep are you producing less because your neurons aren't firing as much, but you also have these other intrinsic mechanisms that clear A beta and just a lot of different metabolites from the brain and proteins, either to the CSF or to the blood, and just this bulk flow process that happens. And if you disrupt sleep, you have less clearance, you're awake more, so you have more production, and again, it promotes a pro-aggregatory state.
So is that it? That explains type 2 diabetes connection. You just have hyperglycemia and you make too much A beta, and maybe you don't sleep as much, and so you don't clear it as well.
I think that there's a lot to do with that. I do think that there's the story of insulin and insulin resistance is one that's unbelievably complex, and I think we're still working that out. But a big component of type 2 diabetes that people don't really, I think, fail to truly understand is the vascular component. When you have type 2 diabetes, you have a lot of vascular damage. And, and vascular impairment is a huge part of Alzheimer's disease. And again, I had mentioned earlier that you kind of have these comorbidities of disease when somebody comes to autopsy, where you're not just a pure Alzheimer's disease brain, but there's another form of dementia called vascular dementia that happens, where hyperglycemia is really toxic for that. So I think that there's a bunch of mechanisms at play, but certainly a sedentary lifestyle, dysregulated metabolism, poor sleep is all bad for the brain in general, and specifically for Alzheimer's.
Why do neurons release A beta? It's bad.
Well, so it's not bad as long as it's cleared, right? Yeah. And so it's really, to this day, we don't know what the function of A beta is. But we do know, so A beta is just a cleavage product, a byproduct of chopping up amyloid precursor protein. And that's a transmembrane protein that's actually found at the synapse that is great for synaptic plasticity and neurotransmission. So it's hard to know whether A beta is just kind of a toxic metabolite that happens, and if there's just too much of it made and we're living too long and then it's aggregating, or whether there's a specific function around A beta that's actually has something that we yet as a field don't fully understand. But definitely APP has a very important function for synaptic homeostasis and synaptic plasticity.
So is it a one-way trip? It's like the glucose metabolism controls Alzheimer's disease. So the direction of cause and effect is not complicated at all.
It's unbelievably complicated because, so you can have the fact that if you're metabolically dysregulated, then that's going to either hasten the onset or move you through the Alzheimer's, the stages of Alzheimer's disease more quickly. But then there's also this piece is if you never have any metabolic problems whatsoever, the mere presence of amyloid pathology seems to cause a metabolic crisis in an individual. And so, we found in our animal models that that's sufficient to produce almost a diabetes-like phenotype where the animals seem like they're insulin resistant, they're not responding to glucose properly, their glucose levels are going higher, they're expending too much energy, but they're eating less. It's a very complex metabolic phenotype, but that's independent of whether the animals ever had a poor diet in their life or type 2 diabetes. There's just something that is very metabolically challenging to have these protein aggregates in the brain that is, counterproductive to healthy aging.
So you see that in experimental animals, but it seems sort of mysterious how it would work.
Yeah.
How does the change in the brain alter the...
So I think if you look at other fields, it's not so terribly mysterious. I just think it hasn't gotten a lot of focus in Alzheimer's disease. Again, I studied lysosomal storage diseases for a long time. These are rare pediatric disorders. And a lot of these disorders actually present with a cachexic or a muscle wasting phenotype. And the idea is you're missing, you can't break down things within your lysosomes. So you get lots of lysosomes. The lysosomes are huge and your cells almost it looks like a foamy cell appearance because your whole cytoplasm of a cell is just full of storage products that can't be broken down. Well, for that cell to survive, to make all of these new lysosomes and keep everything moving forward, it's just energetically demanding. So then almost the organism will just almost feed on itself, for in a pro-survival pathway. And I see this with, across a variety of organisms or different forms of lysosomal storage diseases. The same thing can be said actually for cancer and HIV, where you hear the term cachexia or this wasting that happens. And it's because the disease processes are just a metabolically demanding process. Within the brain, you actually have not only these aggregates, you have to form the aggregates, you have to maintain the health of pre-Madonnas like neurons, but a lot of the glia or other cells in the brain then take on a secondary responsibility. Instead of just helping to support a neuron, they have a pro-inflammatory component now, which is something that, requires energy. So I think that if you look across diseases, you actually see that there's a metabolic tax to not being healthy that then, makes, when you do get a disease, very poor outcomes.
So the way you say it makes it sound sort of global, like if my hippocampus starts to get messed up, then my whole metabolism is going to be messed up. But there's a lot of very local metabolism right around the neurons in the hippocampus, and a lot of what happens in Alzheimer's disease is sort of focused on particular regions in the brain. So is there a feedback mechanism between Alzheimer's pathology in the tissue and the things that control local metabolism in that same tissue?
So I think, so the short answer to that is yes. So there's a lot of work happening right now, again, using amyloid plaques, that what happens right around where a plaque forms is very different, even within, say, a hippocampus that's very vulnerable to disease, or the cortex, that a lot of the activity or the metabolic demand comes where that plaque is formed. It comes back to how all the cells around that plaque respond to it. And that's really what causes this energy sink. But if you even go, a short distance, like 50 microns, 100 microns within the same region, you have a much bigger or different picture of the metabolic milieu than there. The issue is that's just with time, as you get more pro-aggregatory events or more plaques form, then that just takes over the hippocampus in general. But it obviously then has, there's a lot of network disruption that happens in Alzheimer's disease. So the regions that are signaling to one another, of these functional networks, that network connectivity, as we saw it, how connected those networks are, start to become disconnected. And so it then does have impact on other regions that are typically needed for proper brain function.
So sorting out cause and effect isn't just a problem of humans. It's that there are many causes and many effects, and they are only linked to each other loosely, and it makes it very hard, doesn't it? If you want to give a mechanistic explanation for something, you'd like to have a relatively small number of causes and a relatively small number.
Oh yeah, that's not happening in Alzheimer's disease. So I think the field has actually been, somewhat stalled, I think, with therapeutics because we keep looking at, for a long time it was amyloid, and now it's amyloid and tau. But the fact of the matter is that the disease processes are affecting all cell types. It's affecting the brain, it's affecting the periphery. And so again, taking a play out of the cancer playbook where you have, drug intervention, you have surgical intervention, you phenotype the tumor. So the type of approaches you have to treating the disease are based on precision medicine. That's the same type of thing I think that we need to do in Alzheimer's disease. We need to understand that this disorder is unbelievably heterogeneous and that people get Alzheimer's disease for very, very different reasons. They have different comorbidities. Again, if you have type 2 diabetes, cardiovascular disease, and Alzheimer's, then you better believe that the vasculature is going to be a big component of that, which might be different than if you have just an Alzheimer's disease brain. And so I think understanding what the shared mechanisms are, so again, that you can target those effectively, and understanding that it's just going to be multimodal. There's many causes, many things. And I think personally, I try to look how all the things are associated and integrated together. And those commonalities might be something that's a more broad target than coming down to one mechanism alone.
So speaking of that, even A beta, which at one time was thought to be the final common pathway for pathology, now that seems to be at least partly in question.
I think when the amyloid hypothesis first came out in the mid-90s, it was really thought that this was causal for disease. And I think that story has been rewritten. It's not causal, but at this current time, Alzheimer's is diagnosed whether you at autopsy, whether you have changes in amyloid, changes in tau, and neurodegeneration. That is the consensus. But whether those are just a response to other things happening and a biomarker of disease, or actually the one thing that causes the memory impairment, the loss of executive function, I think we've definitely moved away from that hardline approach to all of this.
So if we're sort of thinking about how glucose metabolism alters A beta, and thinking of that as the causal chain to the
a causal change.
Very
many. Yeah, there's many. I would never say it's the causal change. I just think it's one way to accelerate the onset and hasten the amyloid
because
burden.
Glucose metabolism be working on other things as well?
Oh, yeah, absolutely. So, for example, when you all have an insult within your brain, it doesn't matter if it's a traumatic brain injury, MS, Alzheimer's disease, you have a glial response. You have microglia and astrocytes respond to that insult to try and, kind of either quiet the brain down, or if they get a little angry, they can become pro-inflammatory. Well, what we know from a lot of literature in the periphery is that the immune cells are highly glycolytic, right? So glucose can do one thing to the brain or to neurons, but it's going to do something different to, say, the glia, in order to have these dualistic functions to support, but also respond to stimuli, need energy. So I don't think glucose is inherently bad. It's this concept of everything in moderation. And as you start to develop these pathologies associated with Alzheimer's disease, I think you just lose the metabolic flexibility necessary to maintain health and resilience. And that's, I think, more what happens. And glucose is one way that this happens.
Well, as an experimentalist, it almost seems like a minefield to me, because there's so many different things that you could be viewing as causes, and so many different things that you could be viewing as effects. And then every cause could be altering every effect.
That's right.
And so if you imagine the sort of graph of cause and effect, there's about a zillion things here and a zillion things here, and there's a line connecting everything to everything else.
Yeah, but I think that's where you can start to understand, we've, shown that or lots of people have shown that if you have disrupted sleep, it increases Alzheimer's related pathology. If you have Alzheimer's related pathology, it disrupts sleep. So that means that it seems pretty smart to either try to target sleep in order to actually reduce pathology, or vice versa.
When you say target, though, you're thinking of therapy, and I'm just thinking about experimentally figuring out what's going on. Yeah. And so those are two different things, because Right? Because if you just find something that works, you don't have to worry too much about all the weird intricacies of cause and effect.
But again, you can dissect it out, right? So you can ask the question: does amyloid pathology disrupt sleep? So we ask that question. Yes, it does. We also know that if you sleep deprive an animal, it causes A beta levels in the interstitial fluid to go up. So then that identifies. You start to understand it is bidirectional and not unidirectional. That's powerful, right? Because then it's also the idea, if we can, with some of the recent triumphs of some of the anti-amyloid therapeutics, then you can be like, well, this might be some way to actually help restore sleep. So I think it's something that if you just understand the limitations of what you're testing and the fact that they could be bidirectional and have other things impacting it, you can still get the answer that moves the field forward, right? And I think sometimes, the field in some regards for a while was a little too reductionist, just focusing on amyloid, and then it became tau. But now I think there's a lot more that's going into understanding the complexity of it, which then hopefully will lead to a greater knowledge of the disease itself.
So some of the things that you showed in your lecture today seemed like potentially treatments that were interfering with some cause and effect relationship that you'd established. Like one of them was insulin affecting drug.
So metformin was a drug. Yeah. So that and again, there's a lot of drugs out there. So you figure that within the human body, there's a lot of redundant systems, right? It doesn't make sense for every system to just use one molecule that's not found everywhere else. And so there's this idea that if you have these common mechanisms, then if you develop a drug, say for cancer that impacts glucose metabolism, or one for type 2 diabetes, then maybe that could have a different impact. And so we've gone through a lot of different diabetes drugs at this standpoint. Like metformin, it seems to rescue sleep because, again, it restores glucose tolerance. It restores glucose homeostasis within the animal, and that quiets down sleep impairment in mice. But then we have others, that if you look at the drug mechanism, we realize that there's another diabetic drug called glyburide. It's a sulfonylurea that acts on KATP channels. Well, the KATP channels in your pancreas are what secretes insulin, but these channels are also found on the vasculature, and it seems in Alzheimer's disease, due to the amyloid plaques, the vasculature becomes unbelievably rigid and tight. It should be elastic, it should pulse, it should flow, and so you get a phenomenon that's called arterial stiffness. And we find that if you target those KATP channels, it brings their ability to modulate excitability of the vasculature and into a dynamic range, so they can dilate and they can constrict, which is what you need to pump flow. And when you do that, the brain gets happier because they get the goodies it needs. Then you don't have as much stress with this hyperexcitability, and less plaques are made. So I think that there's a lot of ways that if you start to understand and dissect a mechanism, and then you can use a lot of these approaches to target that mechanism, then in combination of those, you might actually have something that could be beneficial.
So these are ideas about how to actually treat Alzheimer's disease. It's always seemed to me that the Alzheimer's disease field has always had a paucity of treatments. There was a lot of ideas about causes, but almost no ideas about treatment. I think a lot of Alzheimer's patients had wished it was the other way around.
Yeah.
And that so it's good to hear. What do you think are the prospects for treatment of Alzheimer's disease based on some of this stuff?
Yeah, so I think one of the things that kind of stymied the field for a while is that there's a couple things. So first off, I think it's really been in the last 5 or 10 years that we fully understood how the pathological changes with time happen in Alzheimer's, and understand what we call the Alzheimer's disease continuum. And I think what's really come out, and again, I would give it maybe the last 5 or 10 years, is that a lot of the changes we see happen so far before anybody shows up with any symptoms of Alzheimer's. And that's what makes this disease so unbelievably challenging, is it's 10, 15, 20 years before somebody has a memory complaint that their amyloid's starting to accumulate, and then later tau and those types of things. And so even when they were developing clinical trials, they were doing it after disease onset. But at that point, we know now that you really can't move, say, amyloid plaque burden, and you need to treat earlier. But because these are diseases of aging, because it takes a long time to change pathology, it's just a numbers game of time, right? I don't think any company or whatever had gone at it ill-informed per se. It's just we learn so much from each trial, and those trials take, with development, treatment, and everything, 5, 10 years, to get the answer that then informs the next trial. And so I think that unlike something like a cancer where you get, you get your answer within a month or with a very hard outcome, right? It's just much different for Alzheimer's. But having said that, I think since we've kind of reshaped how we think about the amyloid hypothesis, we also understand that if you're looking at cognitive changes, it's more related to tau, that we still don't know how amyloid and tau converge to make Alzheimer's, but there's a lot of thoughts on, whether it's neuroinflammation or the vasculature. Now there's a lot of different people companies and researchers looking at those other mechanisms and whether they could be targetable and helpful. The other thing that's come up, I think, that's really blossomed in the last 5 to 10 years is this idea of all the genome-wide association studies that have given genetic risk factors and really looking at a lot of those risk factors to see how that impacts pathology. So for example, with almost all of the genetic risk factors are actually point to monocytes and microglia. They're not actually on the neurons, right? So now a lot of therapies are being directed towards that, or other glia like astrocytes, ApoE, which, is the biggest genetic risk factor. And the idea that, maybe that's something that we should go at and target. And and the biggest pool of ApoE is actually in the periphery. So, can you target something peripheral to then help the brain? So I think by understanding a little bit more about the complexity, not ignoring the complexity of it all, but you're just getting a more, you're storming the castle from different areas in order to hopefully have an effect. But I do think that early trials with amyloid and amyloid antibodies really taught us a lot about the timing that you need to intervene. We also know now how to stage the diseases, all the tracers that could pick up amyloid and tau in the brain. I mean, they're 5, 10, 15 years old. They're new. Same thing with some of the assays that are looking. And the biomarker field that is just blossoming to actually understand a little bit more what's actually changing.
And I think coming forward from so many different points of view, like now that you have these GWAS studies, that can feed into this idea of the, precision medicine. Like let's target, which groups of individuals have these different genetic risks, and then what pathways do they fall into, and what's a good target, treatment for them.
Absolutely. Yeah. And I think that's the biggest thing, is that as we move forward, it's not going to be a one size fits all, right? And if you have, say, really bad, and amyloid within the brain, then maybe an anti-amyloid antibody is not your, not what you should lead with, right? Mm-hmm. So I think, as you said, like really trying to hone in on how to approach the disease more
on an individual basis.
That's absolutely right. Yep.
Also, genetic markers have the advantage of being measurable at any time during life.
That's correct.
You don't have to wait until you're old.
That's right.
Yeah. And ultimately, hopefully, the idea is that you can begin to predict disease risk much earlier, and then, is there something available that's not harmful that you can start taking early on to try and prevent that risk?
Or even lifestyle, modifiers. Yeah, and I think
exercise, more sleep.
That's right. So I think
diet.
Right.
Yeah, like the Mediterranean diet, for instance, which has been known to kind of reduce the risk for Alzheimer's, right? So
Yeah, and I think today the question was raised about the ketogenic diet, right? And so there's a lot with, say, ApoE that intrinsically it seems like your metabolism's a little bit different. And so what might work for an ApoE4 carrier versus an ApoE2 carrier might be very different. But again, if we get that information earlier, then knowing that there's actually literature out there that if you are ApoE4 positive, which can increase your risk up to 12-fold, that if you maintain a healthy diet and exercise very consistently, that you can actually mitigate the genetic risk, right? So there's a lot of things that, with sporadic Alzheimer's disease, you're not destined to get it, right? It's not the end stage of aging. There are things that you can do to minimize your risk. You might not make that risk go away, but it's something that you can focus on to try and mitigate. And it also doesn't have to come from a pill. It just has to, and I think that's the hardest thing for people to hear. And also, it can't happen when you're already diagnosed. It has to happen, in my age range, when you're middle age, right? It's really, you hear a lot about the biology of aging, where as you're young, you build reserve, and then as you're aging, you use up your reserve, and that discrepancy is what's called resilience, right? And so if you can maintain your reserve as you age, so that you don't deplete that reserve, then you'll have more resilience to this disease. And I think that's something to very much consider, as you approach middle age,
That sounds like good advice. So thank you very much, Shannon Macauley and Melanie and Uchit, for joining us today. And this is the last podcast for the semester. We're going to go on Christmas break for a little while. So if you're listening to the podcast, don't think we've given up. We're still here, but we're going to be back in January. This has been Neuroscientists Talk Shop.